Inorganic and Organic Components of Soils, UGC NET JRF Environmental Science Complete Lecture. Soil is a complex, dynamic, and essential natural resource that sustains plant life and influences water movement, air exchange, and the behavior of nutrients in ecosystems. The soil matrix is composed of both inorganic and organic components that contribute to its structure, fertility, and overall functioning. Understanding the composition and roles of these components is vital for the study of soil science and environmental management. This lecture covers the inorganic and organic components of soils, their significance, and their interactions, structured in maximum detail for UGC NET JRF Environmental Science preparation. Inorganic components of soils. The inorganic components of soil primarily consist of minerals, which originate from the weathering of rocks. These minerals are classified into several categories based on their chemical composition and texture. Soil minerals. Sand. Sand particles are the largest inorganic component of soil. They are granular in texture, non-adhesive, and do not hold water or nutrients well. However, they contribute to soil aeration and drainage. The size of sand particles typically ranges from 0.05 mm to 2 mm in diameter. Silt. Silt particles are finer than sand and have a smooth texture. They hold more water and nutrients compared to sand but are still not as effective as clay. Silt particles range in size from 0.002 mm to 0.05 mm. Clay. Clay particles are the finest of all soil particles. They have a sticky texture and can retain large amounts of water and nutrients. Clay particles are smaller than 0.002 mm in diameter. They play a significant role in soil fertility, water retention, and nutrient availability. Soil solution, water and dissolved ions. Soil solution refers to the water held in the pore spaces of the soil, containing dissolved ions such as calcium, Ca², magnesium, Mg², potassium, K, sodium, Na, and sulfate, S². These ions are essential for plant growth and soil chemistry. The composition of soil solution varies with soil type, moisture content, and environmental conditions. The presence of soluble minerals and nutrients in the soil solution is critical for plant nutrition and biological processes. Soil air. Soil air is a mixture of gases, primarily nitrogen N, oxygen O, and carbon dioxide CO found in the pore spaces of the soil. The composition of soil air changes with soil type, moisture, and microbial activity. Oxygen is crucial for root respiration, while CO is a byproduct of microbial and root activity. Soil aeration, which depends on the balance of these gases, is essential for the healthy growth of plants and the functioning of soil organisms. Organic components of soils. The organic components of soil are derived from plant and animal material and contribute to soil fertility, structure, and biological activity. These components primarily consist of organic matter in various stages of decomposition. Soil organic matter. Soil organic matter is a complex mixture of plant and animal residues at different stages of decomposition. SOM is composed of humus, which is a stable, decomposed portion, and the labile fraction, which consists of more easily decomposable plant and animal residues. Humus. Humus is the dark, amorphous organic material in the soil formed by the decomposition of plant and animal residues. It is rich in nutrients and contributes to soil structure by improving its ability to retain water and nutrients. Humus also promotes soil aggregation and enhances soil aeration. It is highly beneficial for soil fertility and supports diverse soil organisms. Decomposing plant and animal residues. These residues include dead plant material, leaves, roots, stems, and animal remains. As they decompose, they release nutrients into the soil, which plants can absorb. The rate of decomposition depends on factors such as temperature, moisture, and the availability of decomposers, e.g., bacteria, fungi. Soil microorganisms. Soil is home to a vast array of microorganisms, including bacteria, fungi, protozoa, and actinomycetes. These microorganisms play a key role in the decomposition of organic matter, nutrient cycling, and the maintenance of soil structure. They break down complex organic compounds into simpler molecules, which plants can absorb. Microbial activity is influenced by soil moisture, temperature, pH, and the availability of organic material. Bacteria. Bacteria are the most abundant microorganisms in soils and are involved in processes like nitrogen fixation, organic matter decomposition, and nutrient cycling. Fungi. Fungi are important decomposers, especially in the breakdown of cellulose and lignin in plant material. They form symbiotic relationships with plant roots, mycorrhizae, to enhance nutrient absorption. Protozoa. Protozoa feed on bacteria and other small organisms, influencing microbial populations and nutrient availability. Earthworms and soil fauna. Earthworms, arthropods, and other soil fauna contribute to the physical and chemical properties of soil by burrowing, mixing, and digesting organic matter. Their activities help in the breakdown of organic material, the formation of soil aggregates, and the improvement of soil structure. Earthworms, in particular, are vital in nutrient cycling, promoting soil fertility, and enhancing water infiltration. Importance of inorganic and organic components. The interaction between inorganic and organic components of soil is crucial for its fertility, structure, and biological activity. The inorganic components sand, silt, clay, minerals, and soil solution provide the physical framework of the soil, determining its texture, drainage capacity, and ability to hold water and nutrients. Organic components SOM, microorganisms, and fauna contribute to soil fertility, nutrient cycling, and the overall health of the soil ecosystem. Soil fertility. The balance between inorganic and organic components affects soil fertility. Soils rich in organic matter tend to have better nutrient and water retention, which supports plant growth. Organic matter also provides a habitat for soil microorganisms, which are essential for nutrient cycling. Soil structure. Organic matter plays a critical role in binding soil particles together to form aggregates, which improve soil structure, aeration, and water infiltration. The interaction between organic matter and clay particles enhances soil aggregation and prevents soil erosion. Nutrient cycling. Inorganic components such as minerals provide essential nutrients to plants, while organic matter contributes to the cycling of nutrients like nitrogen, phosphorus, and carbon. The decomposition of organic matter by soil microorganisms releases nutrients into the soil, which are then taken up by plant roots. Previous year questions with answers. Which of the following is an inorganic component of soil? A. Humus. B. Sand. C. Earthworms. D. Bacteria. Answer. B. Sand. UGC NET 2017. What is the primary role of humus in soil? A. It provides nutrients for plants. B. It improves soil aeration and structure. C. It increases soil temperature. D. It reduces soil erosion. Answer. B. It improves soil aeration and structure. UGC NET 2016. Which of the following microorganisms is most responsible for nitrogen fixation in soil? A. Fungi. B. Protozoa. C. Rhizobium bacteria. D. Actinomycetes. Answer. C. Rhizobium bacteria. UGC NET 2018. What is the primary factor that determines the rate of decomposition of organic matter in soil? A. Soil texture. B. Soil temperature. C. Soil moisture. D. pH. Answer. B. Soil temperature. UGC NET 2020. Which of the following is an example of an organic component in soil? A. Clay. B. Sand. C. Humus. D. Silt. Answer. C. Humus. UGC NET 2019. What role do earthworms play in the soil ecosystem? A. They decompose plant material and improve soil fertility. B. They fix nitrogen in the soil. C. They reduce soil pH. D. They increase the amount of soil erosion. Answer. A. They decompose plant material and improve soil fertility. UGC NET 2021. This lecture has covered both inorganic and organic components of soils, their roles, and their interactions in detail, along with previous year questions to reinforce your preparation for UGC NET JRF Environmental Science. Let me know if you need any further explanations or the next topic. You said. Biogeochemical cycles, nitrogen. ChatGPT said. Biogeochemical cycles, nitrogen cycle, UGC NET JRF Environmental Science Complete Lecture. The nitrogen cycle is one of the essential biogeochemical cycles, playing a crucial role in the environment by cycling nitrogen through various forms between the atmosphere, living organisms, and the Earth's crust. Nitrogen is an essential element for all living organisms as it is a major component of amino acids, proteins, and nucleic acids. The nitrogen cycle ensures the availability of nitrogen in a usable form for plants and animals while maintaining ecological balance. This lecture delves deeply into the nitrogen cycle, focusing on the processes involved, the microorganisms responsible, and the impacts on environmental science. The content is structured with maximum detail for UGC NET JRF environmental science preparation. Key concepts of the nitrogen cycle: atmospheric nitrogen N. Nitrogen makes up about 78% of the Earth's atmosphere, primarily in its inert molecular form N. Despite its abundance, atmospheric nitrogen is not directly usable by most organisms, which is why it needs to be converted into other nitrogenous compounds before it can be assimilated by plants and animals. Nitrogen fixation. Nitrogen fixation is the process by which atmospheric nitrogen N is converted into forms usable by living organisms, primarily ammonia NH or ammonium ions NH. This process can be carried out in two primary ways. Biological nitrogen fixation. Certain bacteria, such as Rhizobium in legume root nodules and Azotobacter, have the enzyme nitrogenase, which can break the strong N as defined as N bond of atmospheric nitrogen and convert it into ammonia. This process is vital for the nitrogen cycle, as it introduces usable nitrogen into the ecosystem. Abiotic nitrogen fixation. Atmospheric nitrogen can also be fixed through abiotic processes, including lightning, which produces nitrogen oxides like NO, or industrial processes like the Haber-Bosch process used to produce fertilizers. Nitrification. Nitrification is a two-step microbial process that converts ammonia or ammonium ions into nitrates, which are highly usable by plants. Ammonia oxidation. The first step is the oxidation of ammonia NH into nitrates by ammonia oxidizing bacteria AOB such as Nitrosomonas. This process requires oxygen. Nitrite oxidation. In the second step, nitrates are further oxidized to nitrates by nitrite oxidizing bacteria such as Nitrobacter. This also occurs in the presence of oxygen. Nitrates are the primary nitrogen form absorbed by plants from the soil, making nitrification a crucial step in the cycle. Assimilation. Assimilation is the process by which plants take up nitrogen in the form of nitrates or ammonium NH from the soil. These forms of nitrogen are then incorporated into amino acids, proteins, and other nitrogen-containing compounds, which are consumed by herbivores and other organisms when they eat plants. Animals obtain nitrogen by consuming plants or other animals, and the nitrogen is used in the formation of proteins and nucleic acids. Ammonification, mineralization. Ammonification is the process by which microorganisms decompose organic nitrogen compounds from dead plants, animals, and animal waste into ammonia NH or ammonium ions NH. This process recycles nitrogen back into the soil in a form that can either be taken up by plants or undergo nitrification. The microorganisms involved in this process include bacteria and fungi that break down organic matter and release ammonia into the soil. Denitrification. Denitrification is the process by which nitrates are reduced to nitrogen gases N or nitrous oxide NO by gentrifying bacteria, such as Pseudomonas and Clostridium, in anaerobic conditions. This process occurs in waterlogged soils, wetlands, and other oxygen-poor environments. Denitrification is crucial because it closes the nitrogen cycle by returning nitrogen back to the atmosphere, maintaining the balance of nitrogen in the ecosystem. Nitrous oxide NO, a byproduct of denitrification, is a potent greenhouse gas, contributing to climate change. Nitrogen cycle diagram. A simplified version of the nitrogen cycle involves the following stages. Nitrogen fixation, atmospheric N right pointing arrow NH or NH. Assimilation, plants absorb NO or NH. Ammonification, organic nitrogen right pointing arrow NH or NH. Nitrification, NH or NH right pointing arrow, no right pointing arrow, no. Denitrification, no right pointing arrow, N or NO. Role of microorganisms in the nitrogen cycle. Microorganisms play a central role in almost all stages of the nitrogen cycle. Nitrogen-fixing bacteria, e.g., Rhizobium, Azotobacter, convert atmospheric N into ammonia, NH, which can be utilized by plants. Ammonia-oxidizing bacteria, e.g., Nitrosomonas, oxidize ammonia to nitrates, no. Nitrite-oxidizing bacteria, e.g., Nitrobacter, oxidize nitrates to nitrates, no, which plants can absorb. Gentrifying bacteria, e.g., Pseudomonas, reduce nitrates back to nitrogen gas, N, completing the cycle. Human impact on the nitrogen cycle. Human activities, especially the use of nitrogen-based fertilizers in agriculture, have significantly altered the nitrogen cycle. The excessive use of fertilizers leads to eutrophication. The runoff of excess nitrates into water bodies causes algal blooms, which deplete oxygen and harm aquatic life. Increased nitrous oxide emissions. Agricultural practices that encourage denitrification contribute to increased emissions of nitrous oxide, NO, a potent greenhouse gas. Soil acidification. Ammonium-based fertilizers can acidify the soil, affecting soil health and nutrient availability. Importance of the nitrogen cycle. The nitrogen cycle is essential for the maintenance of soil fertility, the growth of plants, and the proper functioning of ecosystems. It ensures the availability of nitrogen in a form usable by plants, which, in turn, supports the rest of the food chain. Without this cycle, ecosystems would face a nitrogen deficit, severely affecting life on Earth. Previous year questions with answers. Which of the following is directly responsible for the fixation of atmospheric nitrogen into ammonia? A. Nitrite oxidizing bacteria. B. Gentrifying bacteria. C. Nitrogen fixing bacteria. D. Ammonia oxidizing bacteria. Answer. C. Nitrogen fixing bacteria. UGC NET 2018. In the nitrogen cycle, nitrification is the process of conversion of ammonia into A. Nitrogen gas. B. Nitrites and nitrates. C. Nitrous oxide. D. Ammonium ions. Answer. B. Nitrites and nitrates. UGC NET 2017. Which of the following processes involves the conversion of nitrates into nitrogen gas? A. Nitrogen fixation. B. Denitrification. C. Nitrification. D. Ammonification. Answer. B. Denitrification. UGC NET 2016. The bacteria responsible for converting ammonium ions into nitrates are A. Pseudomonas. B. Nitrobacter. C. Rhizobium. D. Azotobacter. Answer. B. Nitrobacter. UGC NET 2019. What is the main consequence of excessive nitrogen fertilizer use in agriculture? A. Decreased atmospheric nitrogen. B. Increased biodiversity. C. Eutrophication of water bodies. D. Reduction in soil nitrogen. Answer. C. Eutrophication of water bodies. UGC NET 2020. This comprehensive lecture on the nitrogen cycle is designed to give you a detailed understanding of its processes, importance, and human impacts, ensuring that you are thoroughly prepared for the UGC NET JRF environmental science exam. Let me know if you'd like further clarifications or the next topic. You said, carbon, ChatGPT said. Biogeochemical cycles, carbon cycle, UGC NET JRF environmental science complete lecture. The carbon cycle is one of the fundamental biogeochemical cycles that regulate the flow of carbon through Earth's ecosystems. Carbon is an essential element for life, forming the basic building blocks of organic molecules like carbohydrates, proteins, and nucleic acids. The carbon cycle describes the movement of carbon through various components of the Earth system, including the atmosphere, oceans, terrestrial ecosystems, and geological formations. Understanding the carbon cycle is crucial for environmental science, particularly in the context of climate change, as carbon dioxide (CO) is a significant greenhouse gas. This lecture provides an in-depth overview of the carbon cycle, focusing on the processes involved, the role of various ecosystems, and the human impact on the cycle. The information is structured with maximum detail for UGC NET JRF environmental science preparation. Key components of the carbon cycle: atmospheric carbon dioxide (CO). Carbon exists in the atmosphere primarily as carbon dioxide, CO. The concentration of CO in the atmosphere is a critical factor in regulating Earth's temperature. Carbon dioxide is absorbed by plants during photosynthesis and released by animals and plants during respiration. Photosynthesis and primary production. Photosynthesis is the process by which plants, algae, and certain bacteria convert carbon dioxide, CO, and sunlight into glucose, CHO, and oxygen, O. Through this process, carbon is fixed into organic molecules that form the basis of the food chain. Primary producers, plants and phytoplankton, serve as the entry point for carbon into ecosystems. The carbon absorbed during photosynthesis is used to form organic compounds that provide energy for herbivores, which are consumed by carnivores. Respiration. Respiration is the process by which living organisms release energy from organic molecules, primarily glucose, by breaking them down into carbon dioxide, CO, and water, HO. In plants, respiration occurs at night or during periods of low light intensity. In animals, respiration happens continuously. Through cellular respiration, carbon returns to the atmosphere in the form of CO. Decomposition. When organisms die or produce waste, decomposers, bacteria, fungi, and other microorganisms break down their organic matter into simpler compounds, releasing carbon back into the atmosphere as CO through respiration. Decomposition of plant and animal matter also results in the formation of humus, a stable form of organic carbon in soils that is slowly released over time. Carbon sequestration in soils. Carbon can be stored in soils for long periods, especially in ecosystems like forests, wetlands, and peatlands. Organic carbon is stored in the form of plant matter, roots, and organic compounds within the soil matrix. Soil carbon storage is a crucial factor in the global carbon cycle, helping to mitigate the impacts of rising CO levels in the atmosphere. However, soil carbon can be released back into the atmosphere through microbial activity or disturbances like deforestation or land use changes. Oceanic carbon cycle. The oceans play a critical role in the global carbon cycle. Carbon dioxide, CO, from the atmosphere dissolves into the surface waters of the oceans. Phytoplankton in the oceans perform photosynthesis, fixing carbon into organic molecules. Carbon is also stored in the deep ocean in the form of dissolved inorganic carbon, such as bicarbonates, HCO, and as marine sediments. The process of upwelling brings carbon-rich deep water to the surface, where it can be absorbed by marine organisms. The ocean acts as both a carbon sink and a source, depending on the balance of these processes. Fossil fuel formation. Over millions of years, plant and animal remains can become buried and subjected to heat and pressure, eventually forming fossil fuels like coal, oil, and natural gas. These fuels are rich in carbon. The combustion of fossil fuels, e.g., burning coal, oil, and natural gas for energy, releases stored carbon back into the atmosphere as carbon dioxide, CO, significantly impacting the global carbon cycle. This process is one of the primary contributors to the increase in atmospheric CO levels and global warming. Volcanic activity and geological processes. Carbon is stored in carbonate rocks, such as limestone, and in fossil fuels. Over geological timescales, volcanic activity can release carbon dioxide, CO, from these rocks back into the atmosphere. This long-term process is part of the Earth's natural carbon cycle, but human activities have significantly accelerated the release of carbon through fossil fuel burning. The role of humans in the carbon cycle. Human activities have significantly impacted the carbon cycle, particularly through the burning of fossil fuels and land use changes. These activities have disrupted the natural carbon balance, leading to several environmental consequences. Increased carbon emissions. The burning of fossil fuels for energy, transportation, and industrial activities has led to a sharp increase in atmospheric CO levels. This rise in CO is one of the major contributors to climate change. Deforestation. The clearing of forests for agriculture and urban development reduces the ability of ecosystems to act as carbon sinks. Forests play a critical role in absorbing CO during photosynthesis, and deforestation releases stored carbon back into the atmosphere. Ocean acidification. The increased concentration of CO in the atmosphere leads to higher levels of CO dissolving in the oceans. This causes a decrease in ocean pH, resulting in ocean acidification, which affects marine life, particularly organisms that rely on calcium carbonate for their shells, such as corals and shellfish. Carbon cycle feedbacks. The carbon cycle contains several feedback mechanisms that can either amplify or mitigate the impacts of climate change. Positive feedback. As global temperatures rise, carbon is released from permafrost in soils, which can further accelerate warming. For example, the thawing of permafrost releases methane, CH, a potent greenhouse gas, which amplifies the greenhouse effect. Negative feedback. Increased atmospheric CO levels can lead to enhanced plant growth, a phenomenon known as the CO fertilization effect, which can lead to greater carbon sequestration in plants and soils. However, this effect may not be sufficient to counterbalance the rising emissions. Importance of the carbon cycle. The carbon cycle is integral to the functioning of the Earth's ecosystems and climate. It regulates the levels of CO in the atmosphere, which directly influences global temperatures and the carbon balance. The efficient cycling of carbon between the atmosphere, oceans, and terrestrial ecosystems is essential for life as we know it. Disruptions to this cycle, such as those caused by human activities, have profound implications for climate and biodiversity. Previous year questions with answers. Which process in the carbon cycle is responsible for the release of CO into the atmosphere from plants and animals? A. Photosynthesis. B. Respiration. C. Decomposition. D. Carbon sequestration. Answer. B. Respiration. UGC-NET 2018. What is the primary method by which carbon is fixed from the atmosphere into organic molecules? A. Respiration. B. Photosynthesis. C. Decomposition. D. Nitrogen fixation. Answer. B. Photosynthesis. UGC-NET 2017. Which of the following processes contributes to the release of carbon dioxide into the atmosphere from fossil fuels? A. Decomposition. B. Combustion. C. Photosynthesis. D. Respiration. Answer. B. Combustion. UGC-NET 2016. Which human activity has most significantly impacted the carbon cycle by increasing CO levels in the atmosphere? A. Forest conservation. B. Agricultural expansion. C. Burning of fossil fuels. D. Reforestation. Answer. C. Burning of fossil fuels. UGC-NET 2020. What is the role of oceans in the carbon cycle? A. Only a carbon source. B. Only a carbon sink. C. Both a carbon sink and a source. D. Does not participate in the carbon cycle. Answer. C. Both a carbon sink and a source. UGC-NET 2019. This detailed lecture on the carbon cycle equips you with all the necessary information. F. Importance of the carbon cycle. The carbon cycle is integral to the functioning of the Earth's ecosystems and climate. It regulates the levels of CO in the atmosphere, which directly influences global temperatures and the carbon balance. The efficient cycling of carbon between the atmosphere, oceans, and terrestrial ecosystems is essential for life as we know it. Disruptions to this cycle, such as those caused by human activities, have profound implications for climate and biodiversity. Previous year questions with answers. Which process in the carbon cycle is responsible for the release of CO into the atmosphere from plants and animals? A. Photosynthesis. B. Respiration. C. Decomposition. D. Carbon sequestration. Answer. B. Respiration. UGC-NET 2018. What is the primary method by which carbon is fixed from the atmosphere into organic molecules? A. Respiration. B. Photosynthesis. C. Decomposition. D. Nitrogen fixation. Answer. B. Photosynthesis. UGC-NET 2017. Which of the following processes contributes to the release of carbon dioxide into the atmosphere from fossil fuels? A. Decomposition. B. Combustion. C. Photosynthesis. D. Respiration. Answer. B. Combustion. UGC-NET 2016. Which human activity has most significantly impacted the carbon cycle by increasing CO levels in the atmosphere? A. Forest conservation. B. Agricultural expansion. C. Burning of fossil fuels. D. Reforestation. Answer. C. Burning of fossil fuels. UGC-NET 2020. What is the role of oceans in the carbon cycle? A. Only a carbon source. B. Only a carbon sink. C. Both a carbon sink and a source. D. Does not participate in the carbon cycle. Answer. C. Both a carbon sink and a source. UGC-NET 2019. This detailed lecture on the carbon cycle equips you with all the necessary information for UGC NET JRF environmental science preparation. It covers the cycle's components, the human impact, and its role in climate change, providing a strong foundation for your exam. Let me know if you need further explanations or the next topic. You said. Phosphorus and sulfur. ChatGPT said. Biogeochemical cycles, phosphorus and sulfur cycles, UGC NET JRF environmental science complete lecture. The phosphorus and sulfur cycles are two crucial biogeochemical cycles that regulate the movement of phosphorus and sulfur through the Earth's ecosystems. These cycles are essential for sustaining life, as both phosphorus and sulfur are critical elements required for various biological processes. Understanding these cycles is key for environmental science, particularly in the context of nutrient dynamics, ecosystem functioning, and pollution control. This lecture provides a detailed analysis of both the phosphorus and sulfur cycles, their processes, the role of ecosystems, and the impact of human activities. The information is presented with maximum detail for UGC NET JRF environmental science preparation, focusing on critical points relevant for the exam. Phosphorus cycle. Phosphorus is an essential element for all living organisms, primarily forming part of nucleic acids DNA, RNA, ATP, and phospholipids, which are integral to energy transfer and cellular structures. Unlike other major nutrients, phosphorus does not have a significant gaseous phase and mainly exists in the form of phosphate co-cubed in the environment. The phosphorus cycle primarily involves the movement of phosphorus through rocks, water, soil, and living organisms. Key steps in the phosphorus cycle: weathering of rocks. Phosphorus primarily exists in the form of phosphate salts in rocks, mainly as apatite, a mineral form of phosphate. The cycle begins with the weathering of these rocks by physical, chemical, and biological processes, releasing phosphate ions into the soil and water. Uptake by plants. Phosphate in the soil is absorbed by plant roots, where it is incorporated into organic compounds like nucleic acids, ATP, and phospholipids. Plants, especially in their roots, act as the primary consumers of phosphate. Transferred through the food chain. Herbivores obtain phosphorus by consuming plants, and carnivores get phosphorus by eating herbivores or other animals. Phosphorus moves through the food chain in this manner, entering the tissues of all organisms. Decomposition. When plants and animals die or produce waste, decomposers such as bacteria and fungi break down their organic matter. The phosphorus in their tissues is released back into the soil and water as inorganic phosphate. Sedimentation and geological processes. Some of the phosphate that is released through decomposition or runoff from agricultural land is carried by rivers to the ocean. In aquatic environments, phosphorus precipitates out of the water and settles as sediments on the ocean floor. Over geological time, these sediments may become incorporated into new rock formations, completing the cycle. Sedimentary rocks become significant reservoirs of phosphorus in the Earth's crust. Human impact. The phosphorus cycle has been significantly altered by human activities, especially through the use of phosphorus-based fertilizers in agriculture. These fertilizers increase the availability of phosphate in soils, which can lead to eutrophication in aquatic systems when excess phosphorus runs off into water bodies. Eutrophication causes algal blooms, which deplete oxygen levels and harm aquatic life. Sulfur cycle. Sulfur is an essential element for living organisms, being a critical component of amino acids, vitamins, and coenzymes. The sulfur cycle describes the movement of sulfur through the Earth's atmosphere, hydrosphere, lithosphere, and biosphere. Sulfur exists in various forms, including sulfate, C squared, hydrogen sulfide, HS, and sulfur dioxide, SO. Key steps in the sulfur cycle: weathering of sulfide minerals. Sulfur is released from the weathering of sulfide minerals such as pyrite, Fes, found in rocks. Through oxidation processes, sulfur is converted into sulfate ions, C squared, which are released into the soil and water. Uptake by plants. Sulfate ions in the soil are absorbed by plant roots and incorporated into organic sulfur compounds, such as amino acids, cysteine and methionine, which are vital for protein synthesis. Transferred through the food chain. As with phosphorus, sulfur moves through the food chain when herbivores consume plants and carnivores eat herbivores. Sulfur becomes part of the protein structures and other important compounds in animals. Decomposition and release. Upon death and decay of plants and animals, sulfur compounds are released into the soil and water through decomposition. Decomposers break down organic matter and release sulfur back into the environment as hydrogen sulfide, HS, or sulfate, C squared. Volcanic and geothermal emissions. Sulfur is also released into the atmosphere through volcanic activity, where sulfur dioxide, SO, is produced. This gas can then combine with water vapor in the atmosphere to form sulfuric acid, HSO, which falls as acid rain. Geothermal processes, such as hydrothermal vents, also release sulfur compounds into the atmosphere. Human impact. The sulfur cycle is influenced by human activities such as the burning of fossil fuels, which releases large amounts of sulfur dioxide, SO, into the atmosphere. This leads to the formation of acid rain, which can lower the pH of soils and water, damaging ecosystems. The use of sulfur-containing fertilizers and industrial processes also contributes to changes in the sulfur cycle. Interrelationship between phosphorus and sulfur cycles. Both the phosphorus and sulfur cycles share common processes like weathering, biological uptake, decomposition, and sedimentation. However, their impacts on ecosystems are different due to the distinct chemical behaviors of phosphorus and sulfur. For example, phosphorus is mainly transported through soil and water, while sulfur has a significant atmospheric phase and is influenced by volcanic activity. Both cycles are important for nutrient dynamics and ecosystem health. Human interventions, such as the use of fertilizers and industrial pollution, disrupt the natural balance of these cycles, contributing to environmental problems like eutrophication and acid rain. Previous year questions with answers: which of the following is the main source of phosphorus in the soil? A. Atmospheric deposition. B. Weathering of rocks. C. Decomposition of organic matter. D. Nitrogen fixation. Answer: B. Weathering of rocks. UGC NET 2018. In the phosphorus cycle, what form of phosphorus is utilized by plants? A. Phosphate co-cubed. B. Phosphine pH. C. Hydrogen phosphate H2O squared. D. Elemental phosphorus P. Answer: A. Phosphate co-cubed. UGC NET 2017. What is the main form of sulfur found in the atmosphere? A. Hydrogen sulfide HS. B. Sulfur dioxide. So C. Sulfate C squared. D. Sulfuric acid HSO. Answer: B. Sulfur dioxide. So UGC NET 2019. Which human activity has the most significant impact on the sulfur cycle? A. Use of phosphorus-based fertilizers. B. Deforestation. C. Burning of fossil fuels. D. Reforestation. Answer: C. Burning of fossil fuels. UGC NET 2020. Which process in the sulfur cycle contributes to the formation of acid rain? A. Volcanic eruptions. B. Decomposition of sulfur compounds. C. Oxidation of sulfur dioxide. So D. Nitrogen fixation. Answer: C. Oxidation of sulfur dioxide. So UGC NET 2016. This comprehensive lecture on the phosphorus and sulfur cycles provides you with all the essential information needed for UGC NET JRF environmental science preparation. It covers the processes, human impacts, and interrelations of these cycles, ensuring a strong foundation for the exam. Let me know if you need further assistance. You said. Toxic chemicals, pesticides and their classification and effects. ChatGPT said. Toxic chemicals, pesticides and their classification and effects. UGC NET JRF environmental science complete lecture. Pesticides are chemicals or biological agents used to control, destroy, or repel pests, including insects, rodents, weeds, fungi, and bacteria. While pesticides play a significant role in enhancing agricultural productivity by protecting crops from pests and diseases, they can also be toxic to humans, animals, and the environment if misused or overused. Understanding the classification and effects of pesticides is crucial for environmental science, particularly in the context of sustainability, ecological balance, and human health. This lecture provides a detailed analysis of pesticides, their classification, and the adverse effects they have on the environment and human health, tailored specifically for UGC NET JRF environmental science preparation. The lecture is written in maximum detail, with essential concepts and relevant points necessary for the exam. Pesticides, definition and role in agriculture. Pesticides are a broad group of chemicals that are used to control or eliminate pests, such as insects, weeds, Pesticides are a broad group of chemicals that are used to control or eliminate pests, such as insects, weeds, fungi, and rodents. These chemicals are applied in agriculture, public health, and domestic settings to protect crops, livestock, and humans from pests that may cause diseases, reduce crop yields, or pose a health hazard. While pesticides have significantly increased agricultural productivity, they are also associated with various environmental concerns and health risks. Overuse or misuse of pesticides can lead to pesticide resistance, contamination of soil, water, and air, and harm to non-target organisms, including beneficial insects, wildlife, and humans. Classification of pesticides. Pesticides can be classified in several ways, including based on their chemical composition, target pests, or mode of action. The most common classifications include 1. Based on target organism insecticides, target and control insect pests. Example, DDT, malathion, pyrethroids. Herbicides, used to control weeds. Example, glyphosate, atrazine, 2,4-D. Fungicides, used to control fungal diseases. Example, chlorothalonil, mancozeb, carbendazim. Rodenticides, used to control rodents. Example, warfarin, brodifacoum. Bactericides, target harmful bacteria. Example, copper sulfate, streptomycin. Nematicides, used to control nematodes, parasitic worms. Example, phosphoazot, mocap. 2. Based on chemical composition. Organic pesticides, derived from natural sources, including plants or microorganisms. Example, neem oil, pyrethrins, Bacillus thuringiensis Bt. Inorganic pesticides, chemical compounds that do not contain carbon atoms. Example, copper sulfate, arsenic. Synthetic pesticides, man-made chemicals designed to kill or control pests. Example, DDT, chlorpyrifos, atrazine. Biopesticides, derived from natural materials such as animals, plants, or microorganisms. Example, neem extract, Bt. Bacillus thuringiensis Trichoderma spp. 3. Based on mode of action contact pesticides, kill pests upon direct contact. Example, pyrethroids, malathion. Systemic pesticides, absorbed by plants or animals and kill pests that ingest them. Example, imidacloprid, glyphosate. Fumigants, gaseous pesticides that kill pests by being inhaled. Example, methyl bromide, phosphine. Repellents, keep pests away from plants or animals without killing them. Example, DEET, citronella. 4. Based on persistence in environment. Persistent pesticides, these remain active in the environment for a long period. Example, DDT, chlordane. Non-persistent pesticides, break down quickly in the environment. Example, pyrethroids, neem oil. Effects of pesticides. While pesticides are designed to control pests, their misuse and overuse can have various adverse effects on the environment, human health, and non-target organisms. The main effects of pesticides are categorized as toxicological, ecological, and environmental. 1. Environmental impact. Soil contamination. Pesticides can leach into the soil and persist for long periods, harming beneficial soil organisms like earthworms, microbes, and fungi. This can lead to a decline in soil fertility and health. Water pollution. Pesticides can run off from agricultural fields into nearby water bodies, contaminating rivers, lakes, and groundwater. This leads to the death of aquatic organisms, including fish and amphibians, and disrupts aquatic ecosystems. Air pollution. Volatile pesticides can evaporate and enter the atmosphere, contributing to air pollution and affecting air quality. Some pesticides, like DDT, can even travel long distances from their original application site. Loss of biodiversity. Pesticides can affect non-target species, including beneficial insects like bees, butterflies, and ladybugs, which are essential for pollination and pest control. This leads to a loss of biodiversity and disrupts ecosystem services. 2. Human health risks. Acute poisoning. Exposure to high doses of pesticides can lead to acute poisoning, causing symptoms such as nausea, vomiting, dizziness, headaches, and even death. Organophosphates and carbamates are commonly associated with acute poisoning. Chronic health effects. Long-term exposure to pesticides can result in chronic health issues such as cancer. Certain pesticides, such as DDT, lindane, and glyphosate, have been linked to an increased risk of cancers like leukemia, lymphoma, and brain tumors. Endocrine disruption. Pesticides like DDT and atrazine can interfere with the endocrine system, leading to hormone imbalances and reproductive issues. Neurological disorders. Pesticides such as organophosphates and carbamates can damage the nervous system, leading to neurological disorders, including Parkinson's disease and Alzheimer's disease. 3. Development of resistance. Pesticide resistance. Over time, pests can develop resistance to pesticides, making them less effective. This can lead to the need for higher pesticide doses, further increasing the environmental and health risks. Cross-resistance. Some pests may develop resistance to multiple pesticides, making it difficult to control them with conventional methods. Previous year questions with answers. Which of the following is a non-persistent pesticide? A. DDT. B. Pyrethroids. C. Chlordane. D. Lindane. Answer. B. Pyrethroids. UGC-NET 2019. What is the main risk associated with the use of organophosphates as pesticides? A. Acute poisoning. B. Development of pesticide resistance. C. Soil degradation. D. Water contamination. Answer. A. Acute poisoning. UGC-NET 2020. Which pesticide is commonly associated with causing cancer in humans? A. Glyphosate. B. Pyrethrin. C. Methanol. D. Chlorpyrifos. Answer. A. Glyphosate. UGC-NET 2018. Which of the following is true about pesticide resistance? A. It is less common in pests due to pesticide overuse. B. Resistance occurs when pests develop the ability to detoxify or neutralize pesticides. C. Pesticide resistance leads to a decrease in pesticide efficacy. D. All of the above. Answer. D. All of the above. UGC-NET 2017. Which of the following is a biological pesticide? A. Neem oil. B. Glyphosate. C. DDT. D. Malathion. Answer. A. Neem oil. UGC-NET 2016. This comprehensive lecture on pesticides covers their classification, types, and effects in great detail, addressing both the positive and negative impacts on the environment and human health. It includes the key concepts you need to know for UGC-NET JRF environmental science. Let me know if you need further clarification or additional topics. You said, biochemical. Aspects of heavy metals Hg, Cd, Pb, Cr. ChatGPT said. Biochemical aspects of heavy metals Hg, Cd, Pb, Cr. UGC-NET JRF environmental science complete lecture. Heavy metals, including mercury, Hg, cadmium, Cd, lead, Pb, and chromium crossing, are toxic elements that can cause significant environmental and health issues when present in excessive amounts. They enter ecosystems through various anthropogenic activities, such as industrial discharges, mining, agricultural practices, and improper waste disposal. Despite their high toxicity, these metals can accumulate in organisms, leading to bioaccumulation and biomagnification along food chains, ultimately causing harmful effects to human health, wildlife, and ecosystem stability. This lecture explores the biochemical aspects of these metals, including their toxicological effects, mechanisms of action, and their impact on biochemical processes. Mercury, Hg. 1. Toxicological properties. Mercury is a neurotoxin that can accumulate in living organisms over time. It exists in three primary forms: elemental mercury, Hg, inorganic mercury, Hg squared, and organic mercury, methyl mercury, CHHG. Elemental mercury is a liquid at room temperature and can evaporate, leading to inhalation exposure. Inorganic mercury salts can enter the body through ingestion or skin contact, causing kidney damage. Methyl mercury is the most toxic form, primarily found in fish and seafood. It readily crosses the blood-brain barrier and placenta, causing neurological damage, especially in fetuses and young children. 2. Mechanisms of toxicity. Neurotoxicity. Methylmercury binds to sulfhydryl groups in proteins, altering their structure and function. It interferes with neurotransmission and damages the nervous system by disrupting the normal functioning of neurons and synapses. This leads to cognitive impairment, tremors, motor dysfunction, and in severe cases, death. Renal toxicity. Inorganic mercury can accumulate in kidneys, where it interferes with renal enzymes, leading to nephrotoxicity and renal failure. DNA damage. Mercury also has the potential to cause DNA strand breaks, leading to mutagenicity and carcinogenicity in living cells. 3. Biochemical effects. Enzyme inhibition. Mercury binds to thiol groups in enzymes, inhibiting their activity. This interference disrupts various biochemical pathways, including energy production and protein synthesis. Oxidative stress. Mercury exposure increases the production of reactive oxygen species, ROS, leading to oxidative damage in cells. This damage is linked to aging, cancer, and neurodegenerative diseases. Cadmium, CD. 1. Toxicological properties. Cadmium is a highly toxic metal that primarily enters the body through inhalation of industrial fumes, contaminated water, or food. It accumulates in the liver, kidneys, and bones, causing a variety of adverse health effects. Cadmium exposure is often associated with occupational settings, such as battery manufacturing, mining, and tobacco smoking. It is considered a carcinogen and is linked to lung cancer, renal dysfunction, and bone demineralization. 2. Mechanisms of toxicity. Kidney damage. Cadmium has a high affinity for renal tissues, particularly the proximal tubules, where it disrupts the normal function of kidney cells and causes tubular necrosis and renal failure. Osteotoxicity. Cadmium interferes with calcium metabolism in bones, leading to osteoporosis and bone fragility by inhibiting the action of vitamin D and interfering with calcium regulation. Carcinogenic effects. Long-term exposure to cadmium has been linked to lung cancer due to its ability to induce DNA damage and cause mutagenic and clastogenic effects. 3. Biochemical effects. Enzyme inhibition. Cadmium inhibits enzymes involved in essential cellular processes, such as antioxidant defense systems, e.g., superoxide dismutase and glutathione peroxidase, leading to oxidative stress. Disruption of cellular signaling. Cadmium can interfere with calcium signaling, leading to impaired cellular communication and potentially triggering apoptosis and cell death. Lead, Pb. 1. Toxicological properties. Lead is a neurotoxin that affects the central nervous system, CNS, causing a range of neurological effects, especially in children. Lead poisoning can result from lead-based paints, contaminated water, and industrial exposure. Lead exposure is associated with developmental delays, intellectual impairment, and behavioral disorders in children, and can lead to renal failure and hypertension in adults. 2. Mechanisms of toxicity. Neurotoxicity. Lead interferes with the synthesis of neurotransmitters, disrupts synaptic function, and alters the blood-brain barrier. This impairs cognitive function, motor skills, and learning ability in children. Renal toxicity. Lead accumulates in the kidneys, where it disrupts normal renal function, leading to renal tubular damage and, in severe cases, kidney failure. Hemotoxicity. Lead inhibits the heme synthesis pathway, leading to anemia by interfering with the production of hemoglobin. 3. Biochemical effects. Enzyme inhibition. Lead interferes with enzymes involved in heme synthesis, particularly porphobilinogen deaminase, leading to lead-induced anemia. It also inhibits ATPase and calcium ATPase, disrupting cellular metabolism. Oxidative stress. Lead increases the production of reactive oxygen species, ROS, leading to oxidative damage in cellular structures, including lipids, proteins, and DNA. Chromium crossing. 1. Toxicological properties. Chromium exists in two main oxidation states, CR3 and crossing P. CR3 is an essential trace element in the body, involved in glucose metabolism and insulin regulation, but crossing P is highly toxic and carcinogenic. Chromium P compounds are primarily found in industrial waste from processes like tanning, metal plating, and steel production. 2. Mechanisms of toxicity. Carcinogenic effects. Chromium P is a potent carcinogen, capable of causing DNA damage by interacting with the DNA structure and inducing mutations. Long-term exposure to crossing P is linked to lung cancer, skin cancer, and other types of cancer. Renal and liver toxicity. Both crossing 3 and crossing P can accumulate in the kidneys and liver, causing organ damage. CRP is more toxic and can cause necrosis, cell death, and tissue inflammation. 3. Biochemical effects. Enzyme inhibition. Chromium P binds to thiol groups in proteins and enzymes, inhibiting their activity. This leads to disruption of cellular processes such as antioxidant defense, metabolism, and immune function. Oxidative stress. Similar to other heavy metals, CRP induces oxidative stress by increasing ROS production. This oxidative damage contributes to genotoxicity and mutagenicity. Previous year questions with answers. Which of the following heavy metals is most associated with causing neurological damage in humans? A. Mercury. B. Cadmium. C. Lead. D. Chromium. Answer. A. Mercury. UGC-NET 2019. What is the main source of cadmium contamination in the environment? A. Industrial waste. B. Agricultural runoff. C. Automobile exhaust. D. Mining activities. Answer. A. Industrial waste. UGC-NET 2020. Lead poisoning primarily affects which organ in the human body? A. Brain. B. Liver. C. Kidneys. D. Heart. Answer. A. Brain. UGC-NET 2018. Which of the following metals is a known carcinogen? A. Mercury. B. Lead. C. Chromium. D. Cadmium. Answer. C. Chromium. UGC-NET 2020. Which form of mercury is most toxic and accumulates in the food chain? A. Elemental mercury. B. Inorganic mercury. C. Methyl mercury. D. All of the above. Answer. C. Methyl mercury. UGC-NET 2017. This detailed lecture on heavy metals and their biochemical aspects is designed to cover the essential concepts for UGC-NET JRF environmental science. Let me know if you need further clarification on any specific topic or additional lecture material. You said. And metalloids A's, say, CO, O3, ChatGPT said. Biochemical aspects of metalloids A's, say, and gaseous pollutants CO, O, UGC-NET JRF environmental science complete lecture. Metalloids, such as arsenic and selenium, say, and gaseous pollutants like carbon monoxide CO and ozone O play significant roles in environmental toxicity and human health. These elements and gases, though not always classified as heavy metals, have distinct biochemical characteristics and toxicological properties. In this lecture, we will explore the biochemical aspects of arsenic, selenium, and gaseous pollutants like carbon monoxide and ozone, focusing on their toxicological effects, mechanisms of action, and impacts on biochemical processes in living organisms. Arsenic A's. 1. Toxicological properties. Arsenic is a metalloid that occurs naturally in the Earth's crust but is commonly released into the environment through mining, industrial processes, and agricultural activities. Arsenic exists in various oxidation states, with arsenic 3 arsenite and arsenic B arsenate being the most toxic forms. Arsenic contamination is a major concern, particularly in groundwater, where it has caused widespread health issues in regions like Bangladesh and India. 2. Mechanisms of toxicity. Carcinogenicity. Long-term arsenic exposure is a known cause of skin cancer, lung cancer, bladder cancer, and liver cancer. Arsenic induces DNA damage by forming DNA adducts, interfering with DNA repair mechanisms, and causing mutagenic and clastogenic effects. Oxidative stress. Arsenic compounds, particularly arsenite, increase the production of reactive oxygen species, ROS, leading to oxidative damage in lipids, proteins, and DNA. This contributes to cell dysfunction, apoptosis, and genotoxicity. Disruption of cellular processes. Arsenic interferes with cellular signal transduction and enzyme activity, notably inhibiting sulfhydryl-dependent enzymes, which are involved in various cellular pathways, including energy production and antioxidant defense systems. 3. Biochemical effects. Mitochondrial dysfunction. Arsenic targets mitochondria, leading to impaired ATP production and increased cellular stress. This can cause cell death and contribute to systemic diseases like cardiovascular disease and diabetes. Immunotoxicity. Arsenic suppresses immune system function, making individuals more susceptible to infections and possibly contributing to autoimmune diseases. Selenium, say 1. Toxicological properties. Selenium is an essential trace element in small quantities but becomes toxic at higher concentrations. It is found naturally in the Earth's crust, especially in coal, soil, and water. Selenium is present in various chemical forms, including selenite CO2 and selenate CO2, both of which can be toxic at elevated levels. While selenium is beneficial to health in trace amounts, acting as an essential cofactor in enzymes like glutathione peroxidase, selenium toxicity can occur from industrial emissions, mining, or the consumption of selenium-rich foods. 2. Mechanisms of toxicity. Disruption of antioxidant systems. Selenium in excess can inhibit antioxidant enzymes, particularly glutathione peroxidase, leading to oxidative stress and cellular damage. Protein dysfunction. Selenium competes with sulfur in amino acids, leading to altered protein structure and enzyme dysfunction. This can impair cell signaling, gene expression, and overall cellular function. Neurotoxicity. Chronic selenium exposure is linked to neurological impairments. It disrupts the central nervous system, CNS, leading to symptoms such as mental confusion, fatigue, and neuropathies. 3. Biochemical effects. Genotoxicity. High levels of selenium induce DNA strand breaks, leading to mutations and genetic instability. These effects contribute to cancer development in various tissues. Endocrine disruption. Selenium interferes with thyroid hormone synthesis, affecting metabolic processes and leading to endocrine imbalance. Carbon monoxide CO1. Toxicological properties. Carbon monoxide CO is a colorless, odorless, and tasteless gas that is primarily produced by the incomplete combustion of carbon-containing fuels, e.g., in motor vehicle exhaust, fireplaces, and gas stoves. CO binds to hemoglobin with much greater affinity than oxygen, about 200 to 250 times, forming carboxyhemoglobin (CoHV) and reducing the oxygen-carrying capacity of the blood. 2. Mechanisms of toxicity. Hypoxia. Since CO binds to hemoglobin more effectively than oxygen, it reduces oxygen delivery to tissues, leading to hypoxia, lack of oxygen. This can result in symptoms such as headaches, dizziness, fatigue, and at higher concentrations, loss of consciousness or death. Neurotoxicity. CO affects the central nervous system, CNS, by interfering with neurological function, causing cognitive impairment, motor dysfunction, and long-term neurological deficits. Cellular dysfunction. CO disrupts mitochondrial function by binding to cytochrome C oxidase, an enzyme involved in oxidative phosphorylation, leading to decreased ATP production and increased lactic acid production. 3. Biochemical effects. Oxidative stress. CO exposure increases the production of reactive oxygen species, ROS, leading to cellular damage, particularly in the brain and heart. This oxidative stress can result in inflammation, cell death, and long-term tissue damage. Mitochondrial dysfunction. CO inhibits the electron transport chain in mitochondria, decreasing ATP synthesis and increasing lactic acidosis. Ozone O. 1. Toxicological properties. Ozone O is a highly reactive gas formed by the interaction of sunlight with airborne pollutants. While it plays an essential role in blocking ultraviolet UV radiation in the upper atmosphere, at ground level, it is a major air pollutant and component of smog. Ozone exposure occurs primarily through inhalation of polluted air and can lead to respiratory issues and systemic toxicity. 2. Mechanisms of toxicity. Respiratory toxicity. Ozone causes direct damage to the respiratory epithelium, leading to inflammation, bronchoconstriction, and increased mucus secretion. This leads to asthma, chronic bronchitis, and emphysema. Cellular damage. Ozone induces oxidative stress by increasing the production of reactive oxygen species, ROS. This causes cellular membrane damage, lipid peroxidation, and protein degradation in lung tissues. 3. Biochemical effects. Inflammatory response. Ozone exposure leads to the activation of pro-inflammatory cytokines and the recruitment of immune cells such as neutrophils and macrophages, exacerbating lung tissue damage. Gene expression alterations. Ozone affects gene expression, leading to the upregulation of genes associated with inflammation, oxidative stress, and cell death. Previous year questions with answers. Which of the following metals is most likely to cause lung cancer when exposed to long-term inhalation? A. Mercury. B. Cadmium. C. Arsenic. D. Lead. Answer. C. Arsenic. UGC-NET 2020. Which gas binds to hemoglobin more readily than oxygen and is responsible for carbon monoxide poisoning? A. Nitrogen. B. Oxygen. C. Carbon monoxide. D. Methane. Answer. C. Carbon monoxide. UGC-NET 2019. What is the primary environmental source of ozone formation? A. Industrial emissions. B. Combustion engines. C. Photochemical reactions. D. Forest fires. Answer. C. Photochemical reactions. UGC-NET 2018. Which element, when present in high concentrations, disrupts antioxidant enzymes and causes oxidative stress? A. Selenium. B. Lead. C. Arsenic. D. Cadmium. Answer. A. Selenium. UGC-NET 2021. What is the major mechanism of toxicity of ozone exposure in humans? A. Carcinogenicity. B. Respiratory inflammation. C. Neurological impairment. D. Renal toxicity. Answer. B. Respiratory inflammation. UGC-NET 2022. This detailed lecture on metalloids and gaseous pollutants is designed for UGC-NET JRF Environmental Science and provides in-depth insights into the biochemical toxicity and health effects of arsenic, selenium, carbon monoxide, and ozone. Let me know if you need further elaboration or additional study material. You said, Pan, VOC and Pop. ChatGPT said. Pan, VOC, and Pop, UGC-NET JRF Environmental Science Complete Lecture. Pan peroxyacetyl nitrate, VOC, volatile organic compounds, and Pop persistent organic pollutants are critical environmental pollutants that contribute significantly to atmospheric pollution, human health risks, and ecological damage. These substances, though different in their chemical properties, are linked through their roles in atmospheric chemistry, the formation of secondary pollutants, and their persistence in the environment. In this lecture, we will examine the biochemical aspects, sources, mechanisms of action, and effects of Pan, VOC, and Pop, and provide insights into their environmental significance. Peroxyacetyl nitrate, Pan 1, chemical properties and formation. Pan is a secondary pollutant formed in the atmosphere through a complex reaction involving volatile organic compounds, VOX, and nitrogen oxides, NOx, in the presence of sunlight. Pan primarily forms in urban and suburban areas with high levels of traffic and industrial emissions, where VOX react with NOx to form ozone O, which then catalyzes the formation of Pan. Pan is a reactive compound, known for its ability to cause respiratory distress and eye irritation. 2. Mechanism of toxicity. Respiratory irritation. Pan is highly reactive and, when inhaled, can cause significant irritation to the respiratory tract, leading to symptoms such as coughing, wheezing, and shortness of breath. Prolonged exposure can lead to chronic respiratory conditions such as asthma and bronchitis. Ozone formation. Pan is a contributor to tropospheric ozone formation, which is associated with photochemical smog. Ozone can exacerbate existing lung diseases and impair lung function, leading to severe health consequences over time. 3. Environmental and ecological effects. Damage to vegetation. PAN has been shown to cause leaf injury in plants, resulting in chlorosis, necrosis, and stunted growth. This can have significant effects on agriculture and natural ecosystems. Contribution to air pollution. As a secondary pollutant, Pan contributes to the formation of smog and particulate matter PM2.5, both of which have serious environmental and human health implications. Volatile organic compounds, VOX 1. Chemical properties and sources. VOX are a diverse group of organic chemicals that have a high vapor pressure at room temperature, meaning they can easily evaporate into the air. They are primarily emitted by vehicle exhaust, industrial activities, biomass burning, solvent use, and agriculture. Common VOCs include methane, benzene, formaldehyde, acetone, and toluene, which are present in both indoor and outdoor air. 2. Mechanism of toxicity. Ozone formation. VOCs play a crucial role in the formation of tropospheric ozone, a potent oxidant. In the presence of sunlight, VOCs react with nitrogen oxides, NOx, leading to the production of ozone. This process contributes significantly to photochemical smog. Health impacts. Short-term exposure to VOCs can cause headaches, dizziness, nausea, and eye irritation. Long-term exposure to certain VOCs, such as benzene and formaldehyde, is linked to cancer, liver damage, and neurological disorders. 3. Environmental and ecological effects. Air pollution. VOCs are key contributors to the formation of smog and particulate matter in urban areas. In addition to ozone, VOCs can contribute to the formation of other secondary pollutants, such as pan and peroxy radicals, which exacerbate air quality problems. Ecological impact. VOCs can affect plant metabolism by disrupting photosynthesis and growth. They may also affect soil chemistry, altering nutrient cycles and microbial activity in ecosystems. Persistent organic pollutants, POPs. 1. Chemical properties and sources. POPs are a group of organic chemicals that are characterized by their persistence in the environment, bioaccumulation in living organisms, and toxicity. These substances have long half-lives, and they do not easily degrade in the environment, which makes them a significant concern for environmental and public health. Common POPs include DDT, dichlorodiphenoyl trichloroethane, PCBs, polychlorinated biphenyls, dioxins, and furans. These chemicals are primarily pesticides, industrial chemicals, and byproducts of industrial processes like waste incineration. 2. Mechanism of toxicity. Endocrine disruption. Many POPs act as endocrine disruptors, meaning they interfere with the hormonal system by mimicking or blocking the action of natural hormones. This can lead to reproductive problems, immune system suppression, and developmental issues in wildlife and humans. Carcinogenicity. Many POPs are classified as carcinogens. For example, PCBs and dioxins are known to increase the risk of developing cancers, particularly of the liver, skin, and reproductive organs. Neurological effects. POPs, particularly dioxins and PCBs, have been shown to negatively affect the nervous system, leading to developmental and cognitive impairments, particularly in children exposed to these chemicals during pregnancy or early life. 3. Environmental and ecological effects. Bioaccumulation. POPs accumulate in the food chain, becoming more concentrated as they move up the trophic levels. This leads to high concentrations in top predators, including humans. Global distribution. Due to their persistent nature and the ability to travel long distances, POPs are found in remote areas such as the Arctic, where they have never been used. This global distribution highlights the widespread environmental threat posed by POPs. Ecotoxicity. POPs can significantly affect biodiversity by disrupting reproductive processes, growth, and development in wildlife. For example, the presence of DDT has caused the thinning of eggshells in birds of prey, leading to population declines. Previous year questions with answers. Which of the following is a major contributor to the formation of PAN in the atmosphere? A. Methane. B. Nitrogen oxides, NOx. C. Carbon monoxide. D. Sulfur dioxide. Answer. B. Nitrogen oxides, NOx. UGC-NET 2020. What is the primary health effect associated with the exposure to volatile organic compounds? Box. A. Skin rashes. B. Respiratory issues. C. Bone disorders. D. Liver toxicity. Answer. B. Respiratory issues. UGC-NET 2019. Which of the following pollutants is classified as a persistent organic pollutant? POP. A. Carbon dioxide. B. DDT. C. Sulfur dioxide. D. Nitrogen oxide. Answer. B. DDT. UGC-NET 2021. The formation of ozone O in the atmosphere is primarily driven by the reaction between VOC and which of the following? A. Carbon dioxide. B. Nitrogen oxides, NOx. C. Water vapor. D. Sulfur oxides. Answer. B. Nitrogen oxides, NOx. UGC-NET 2018. Which of the following is not a characteristic of persistent organic pollutants? POPs. A. Bioaccumulation. B. High volatility. C. Persistence in the environment. D. Toxicity to wildlife. Answer. B. High volatility. UGC-NET 2020. This detailed lecture on PAN, VOC, and POP provides a comprehensive understanding of these critical environmental pollutants, their sources, mechanisms of toxicity, and ecological impacts. The included previous year questions will help reinforce the understanding of these topics and guide preparation for the UGC-NET JRF Environmental Science exam. Let me know if you need further elaboration or more study material. You said. Carcinogens in the air. ChatGPT said. Carcinogens in the air. UGC-NET JRF Environmental Science complete lecture. Air pollution is one of the most pressing environmental issues, and among its many hazards, carcinogens in the air pose a significant risk to human health. These carcinogenic substances, when inhaled or absorbed through the skin, can cause cancer or promote the development of cancerous cells in the body. In this lecture, we will explore the sources, types, mechanisms of action, and health effects of carcinogens found in the air. This understanding is crucial for UGC-NET JRF environmental science preparation, particularly regarding air pollution and public health. Types of carcinogens in the air. 1. Particulate matter PM. Particulate matter PM refers to a mixture of tiny particles and liquid droplets suspended in the air. PM can be categorized into different sizes. PM10 particles with a diameter less than 10 micrometers and PM2.5 particles with a diameter less than 2.5 micrometers. Carcinogenic components of PM. Polycyclic aromatic hydrocarbons PAHs, benzene, and arsenic are common carcinogenic compounds found in particulate matter. These substances are known to cause lung cancer, skin cancer, and bladder cancer. 2. Benzene. Benzene is a volatile organic compound, VOC, and a known human carcinogen, primarily found in the air due to emissions from industrial processes, vehicle exhaust, and tobacco smoke. Carcinogenic mechanism. Benzene is metabolized in the body into several reactive metabolites, including benzoquinone, which can bind to DNA and cause mutations, leading to leukemia and other blood cancers. 3. Formaldehyde. Formaldehyde is a colorless gas with a strong odor, commonly released from vehicle emissions, smoking, wood burning, and construction materials, e.g., plywood, insulation. Carcinogenic mechanism. Exposure to formaldehyde can lead to DNA damage, chromosomal aberrations, and mutagenesis, promoting the development of cancers such as nasal cancer, pharyngeal cancer, and laryngeal cancer. 4. Asbestos. Asbestos fibers, when inhaled, can lodge in the lungs and cause serious diseases such as mesothelioma and lung cancer. Though its use has declined, asbestos still remains a risk in older buildings and environments where renovation or demolition activities take place. 5. Radon. Radon is a naturally occurring radioactive gas that seeps into buildings from the soil, particularly in areas with high levels of uranium in the ground. It is the second leading cause of lung cancer after smoking. Carcinogenic mechanism. Radon decay products emit alpha radiation, which can damage lung cells, leading to mutations in the development of lung cancer. Radon is a naturally occurring radioactive gas that seeps into buildings from the soil, particularly in areas with high levels of uranium in the ground. It is the second leading cause of lung cancer after smoking. Carcinogenic mechanism. Radon decay products emit alpha radiation, which can damage lung cells, leading to mutations in the development of lung cancer. 6. Diesel exhaust. Diesel exhaust contains a mixture of gases and particulate matter, including PAHs and nitrogen oxides, NOx. Long-term exposure to diesel exhaust is linked to lung cancer and bladder cancer. The International Agency for Research on Cancer, IARC, has classified diesel exhaust as a group 1 carcinogen, meaning there is enough evidence to conclude that it can cause cancer in humans. Sources of carcinogens in the air. 1. Vehicle emissions. Automobile exhaust contains a cocktail of harmful chemicals, including benzene, formaldehyde, carbon monoxide, nitrogen oxides, and PAHs. These emissions contribute significantly to urban air pollution and the associated health risks. 2. Industrial processes. Certain industrial activities, such as chemical manufacturing, refining, and waste incineration, release toxic substances such as benzene, toluene, formaldehyde, and PAHs into the atmosphere. 3. Tobacco smoke. Cigarette smoke is a significant source of carcinogens like benzene, formaldehyde, and PAHs, which contribute to the development of cancers such as lung cancer, oral cancer, and throat cancer. 4. Residential heating and cooking. The use of wood and coal for heating and cooking releases PAHs, benzene, and other toxic substances that are inhaled by household members. Indoor air pollution from these sources has been linked to lung cancer, particularly in developing countries where the use of solid fuels is common. 5. Biomass burning. In many rural areas, biomass burning, such as burning of wood, crop residue, and other organic matter, is a common source of particular. The use of wood and coal for heating and cooking releases PAHs, benzene, and other toxic substances that are inhaled by household members. Indoor air pollution from these sources has been linked to lung cancer, particularly in developing countries where the use of solid fuels is common. 5. Biomass burning. In many rural areas, biomass burning, such as burning of wood, crop residue, and other organic matter, is a common source of particulate matter, carbon monoxide, benzene, and PAHs, all of which have carcinogenic properties. 6. Natural sources. Radon is a naturally occurring carcinogen that seeps from rocks and soil, particularly in regions with uranium-rich deposits. Radon exposure is a major risk factor for lung cancer in non-smokers. Mechanism of carcinogenicity. The carcinogenic effects of air pollutants are primarily related to their ability to cause DNA damage, mutagenesis, and disruption of cellular mechanisms. Here's a breakdown of how air carcinogens affect human health. DNA damage. Many carcinogenic chemicals, such as benzene, formaldehyde, and PAHs, combine to DNA, forming adducts that disrupt normal cellular function. This can lead to mutations, chromosomal aberrations, and ultimately, cancer. Oxidative stress. Many air pollutants induce oxidative stress by generating free radicals. These highly reactive molecules can damage cell membranes, proteins, and DNA, increasing the likelihood of cancerous transformations. Inflammatory response. Long-term exposure to carcinogens can trigger chronic inflammation, which can alter the normal growth regulation of cells and contribute to cancer development. Endocrine disruption. Carcinogens such as PCBs and DDT can mimic or block the action of hormones, leading to reproductive cancers, e.g., breast, prostate. Health effects of carcinogens in the air. The effects of carcinogens in the air on human health can range from acute symptoms to long-term diseases. Respiratory diseases. Prolonged exposure to carcinogenic air pollutants like PM2.5, formaldehyde, and benzene can lead to chronic respiratory diseases such as bronchitis, asthma, and emphysema, in addition to lung cancer. Lung cancer. The most significant health effect of air carcinogens is lung cancer, particularly from radon, diesel exhaust, and cigarette smoke. Other cancers. Prolonged exposure to benzene and PAHs has been linked to bladder, liver, and kidney cancers. Previous year questions with answers. Which of the following is a major source of benzene in the air? A. Industrial emissions. B. Cigarette smoke. C. Vehicle exhaust. D. All of the above. Answer. D. All of the above. UGC NET 2020. What is the primary health effect associated with long-term exposure to radon? A. Liver damage. B. Lung cancer. C. Skin irritation. D. Heart disease. Answer. B. Lung cancer. UGC NET 2019. Which of the following pollutants is considered a group 1 carcinogen by the IARC? A. Diesel exhaust. B. Ozone. C. Carbon dioxide. D. Sulfur dioxide. Answer. A. Diesel exhaust. UGC NET 2021. Which of the following is a common source of particulate matter PM in the air? A. Vehicle exhaust. B. Biomass burning. C. Industrial processes. D. All of the above. Answer. D. All of the above. UGC NET 2018. Which of the following chemicals is a known human carcinogen found in vehicle exhaust? A. Benzene. B. Methane. C. Nitrogen oxides. D. Carbon monoxide. Answer. A. Benzene. UGC NET 2020. This lecture on carcinogens in the air provides comprehensive insights into the sources, health effects, and biochemical mechanisms of action of carcinogenic air pollutants. The detailed discussion in previous year questions will help solidify your understanding and aid in preparation for the UGC NET JRF Environmental Science exam. Let me know if you need more elaboration on any topic. You said. Principles of analytical methods. Titron tree. ChatGPT said. Principles of analytical methods. Titron tree. UGC NET JRF Environmental Science lecture. Titron tree, also known as volumetric analysis, is a widely used technique in analytical chemistry to determine the concentration of a substance in a solution. This method relies on a chemical reaction between the analyte substance whose concentration is to be determined and a reagent of known concentration titrant. Titron tree is a fundamental technique not only in environmental science for water and air quality analysis but also in various chemical and biological investigations. In this lecture, we will delve into the principles of titron tree, the various types of titrations, and how they are applied in the environmental sciences. The goal is to provide you with an in-depth understanding of titron methods, particularly for the UGC NET JRF Environmental Science exam. Basic principles of titron tree. Titron tree involves the addition of a titrant, a solution of known concentration, to a sample solution analyte until the reaction between the two is complete. The point at which the reaction is complete is called the endpoint. The volume of the titrant required to reach the endpoint is then used to calculate the concentration of the analyte in the sample. Key steps in titron tree analysis. Preparation of the titrant. A solution of known concentration, standard solution, is prepared for the titration. Addition of titrant to sample. The titrant is slowly added to the analyte solution until the reaction reaches the endpoint. Indicator use. An indicator may be added to the analyte solution, which will signal the endpoint by changing color, or the endpoint may be detected through pH changes, electrochemical measurements, or conductivity changes. The calculation of the analyte concentration uses the relationship between the volume of titrant used, its concentration, and the volume of the sample. The formula for concentration calculation is C1V1 equals C2V2 C1V1 equals C2V2. Where C1C1 equals concentration of the titrant. V1V1 equals volume of the titrant used. C2C2 equals concentration of the analyte. V2V2 equals volume of the sample. Types of titron tree. There are several types of titron tree, each suited for different types of chemical reactions. The most common are acid-base titrations, precipitation titrations, redox titrations, and complexometric titrations. 1. Acid-base titration. Principle. In an acid-base titration, the analyte, either an acid or a base, reacts with a titrant of opposite nature, base or acid, respectively. Indicator. A pH indicator such as phenolphthalein for strong acid strong base titration, or methyl orange for weak acid strong base titration, is commonly used to identify the endpoint of the titration. Applications. Determining the acidity or alkalinity of solutions, such as in water treatment, environmental analysis of soil, or acid rain analysis. For example, in a titration between hydrochloric acid HCl and sodium hydroxide NaO. HCl plus. NaO right pointing arrow. NaCl plus. H2O. HCl plus NaO right pointing arrow NaCl plus H2. O. 2. Precipitation titration. Principle. Precipitation titrations involve the formation of a precipitate during the reaction between the analyte and the titrant. Indicator. Fluorescein or potassium chromate is commonly used to detect the endpoint when a precipitate of an insoluble compound forms. Applications used in determining the concentration of ions like chlorides, sulfates, or phosphates in water. For example, when silver nitrate AgNO reacts with chloride ions Cl in water, silver chloride AgCl precipitates. AgNO 3 plus. NaCl right pointing arrow. AgCl S plus. Nano 3. AgNO 3 plus NaCl right pointing arrow AgCl S plus Nano 3. 3. Redox titration. Principle. In redox titrations, the analyte and titrant undergo an oxidation reduction reaction, where one substance is oxidized and the other is reduced. Indicator. The redox potential or indicator solution, e.g., ferric ion indicator of potassium permanganate, can be used to monitor the endpoint. Applications. Used to measure the concentration of oxidizing or reducing agents, such as dissolved oxygen, chlorine, or iron in water samples. An example of a redox reaction is the titration of potassium permanganate KMNO with oxalic acid. MnO4 minus plus C2O4 2 minus right pointing arrow Mn2 plus CO2 MnO4 minus plus C2O4 2 minus. Right pointing arrow Mn2 plus CO2. 4. Complexometric titration. Principle. Complexometric titrations are based on the formation of complexes between metal ions and ligands. EDTA ethylene diamine tetracetic acid is the most common titrant used in complexometric titrations. Indicator. Areochrome black tea is commonly used as an indicator for titrations involving calcium or magnesium ions. Applications. Used for the analysis of water hardness, metal ions, and trace elements in environmental samples. For example, in the titration of calcium ions with EDTA. Ca2++. EDTA4. Minus right pointing arrow. CaEDTA2. Minus Ca2+. Plus EDTA4 minus. Right pointing arrow CaEDTA2 minus. Applications of titrantry in environmental science. Titrantry plays a vital role in environmental science, particularly in water quality analysis, soil analysis, and air quality monitoring. Some applications include determining water quality. Acid-based titrations are used to measure the alkalinity and acidity of water, which are important indicators of water quality and pollution. Hardness of water. Complexometric titrations are commonly used to determine the hardness of water, which is caused by the presence of calcium Ca2 and magnesium Mg2 ions. Dissolved oxygen. Redox titrations help in measuring dissolved oxygen levels in water, a key parameter in assessing the health of aquatic ecosystems. Chlorine content. Chlorine levels in drinking water and wastewater treatment are often measured using redox titrations, with sodium thiosulfate as a titrant. Environmental pollution monitoring. Titration methods can be used to monitor pollutants such as sulfates, nitrates, and phosphates in water bodies. Previous year questions with answers. Which of the following is the most commonly used reagent in a complexometric titration? A. Potassium permanganate. B. Sodium thiosulfate. C. EDTA. D. Silver nitrate. Answer. C. EDTA. UGC-NET 2019. In an acid-based titration, which indicator would be used to detect the endpoint of a strong acid-strong base titration? A. Methyl orange. B. Phenolphthalein. C. Bromothymol blue. D. Litmus. Answer. B. Phenolphthalein. UGC-NET 2020. Which of the following methods is most appropriate for determining the hardness of water? A. Redox titration. B. Complexometric titration. C. Acid-based titration. D. Precipitation titration. Answer. B. Complexometric titration. UGC-NET 2018. What is the purpose of adding an indicator in a titration? A. To change the color at the endpoint. B. To increase the rate of the reaction. C. To determine the concentration of the analyte. D. To act as a buffer. Answer. A. To change the color at the endpoint. UGC-NET 2019. In a redox titration, which substance is commonly used as a titrant when measuring chlorine content in water? A. Sodium thiosulfate. B. Potassium permanganate. C. Sodium hydroxide. D. Silver nitrate. Answer. A. Sodium thiosulfate. UGC-NET 2020. Conclusion. Titrantry is a highly effective analytical method used in environmental science to determine the concentration of various substances in air, water, and soil. Which of the following is the most commonly used reagent in a complexometric titration? A. Potassium permanganate. B. Sodium thiosulfate. C. EDTA. D. Silver nitrate. Answer. C. EDTA. UGC-NET 2019. In an acid-based titration, which indicator would be used to detect the endpoint of a strong acid-strong base titration? A. Methyl orange. B. Phenolphthalein. C. Bromothymol blue. D. Litmus. Answer. B. Phenolphthalein. UGC-NET 2020. Which of the following methods is most appropriate for determining the hardness of water? A. Redox titration. B. Complexometric titration. C. Acid-based titration. D. Precipitation titration. Answer. B. Complexometric titration. UGC-NET 2018. What is the purpose of adding an indicator in a titration? A. To change the color at the endpoint. B. To increase the rate of the reaction. C. To determine the concentration of the analyte. D. To act as a buffer. Answer. A. To change the color at the endpoint. UGC-NET 2019. In a redox titration, which substance is commonly used as a titrant when measuring chlorine content in water? A. Sodium thiosulfate. B. Potassium permanganate. C. Sodium hydroxide. D. Silver nitrate. Answer. A. Sodium thiosulfate. UGC-NET 2020. Conclusion. Titrantry is a highly effective analytical method used in environmental science to determine the concentration of various substances in air, water, and soil. Understanding the principles and different types of titrations, along with their applications, will greatly enhance your knowledge for the UGC-NET JRF environmental science exam. This lecture covers the key aspects of titrantric analysis, providing the foundation for more advanced studies in analytical chemistry and environmental monitoring. You said, gravimetry. ChatGPT said. Gravimetry, UGC-NET JRF environmental science lecture. Gravimetry is a widely used quantitative analysis technique in environmental science and chemistry that involves the measurement of the mass of a substance in a sample. It is one of the most accurate and precise methods for determining the concentration of a substance, particularly when the analyte can be converted into a stable, easily weighed form. The principle of gravimetry is based on the fact that mass is a fundamental property that can be measured with high precision, and it provides a direct means of determining the amount of the substance of interest in the sample. In this lecture, we will cover the principles of gravimetry, the steps involved in the gravimetric analysis process, the common techniques used in gravimetry, and its application in environmental science. This lecture is aimed to provide you with a deep understanding of gravimetry as it applies to the analysis of environmental samples. Principles of gravimetry. Gravimetric analysis involves the separation of a substance from a sample, its transformation into a stable form, and its subsequent weighing to determine the amount present in the sample. The key principle is that mass is a fundamental property that is conserved during the chemical transformations involved in the analysis. Key steps in gravimetric analysis. Sample preparation. The sample is first prepared and, if necessary, dissolved in a suitable solvent. Precipitation. The analyte is converted into a solid, usually by adding a reagent that reacts with the analyte to form an insoluble compound. Filtration. The precipitate is separated from the liquid by filtration. Washing. The precipitate is washed to remove impurities and unwanted soluble components. Drying. The precipitate is dried or ignited to remove any moisture or volatile substances. Weighing. The final dry precipitate is weighed, and its mass is used to calculate the concentration of the analyte in the sample. Gravimetry is particularly useful when the analyte is present in very low concentrations, and it provides highly accurate and reproducible results. Common gravimetric techniques. There are various gravimetric methods used depending on the nature of the analyte and the available reagents. The most common techniques are 1. Precipitation gravimetry. In precipitation gravimetry, a substance, typically an ion, in the sample reacts with a reagent to form an insoluble precipitate. The precipitate is then isolated, washed, dried, and weighed. The mass of the precipitate is used to calculate the concentration of the analyte. For example, when determining the concentration of chloride ions in a water sample, silver nitrate AgNO is added to the sample, forming silver chloride AgCl. Ag plus Cl minus right pointing arrow. AgCl S Ag. Plus Cl minus. Right pointing arrow AgCl S. After filtration, washing, and drying, the mass of the AgCl precipitate is measured, and the concentration of chloride ions is calculated based on the stoichiometry of the reaction. 2. Volatilization gravimetry. Volatilization gravimetry involves heating the sample to volatilize a component, such as water or carbon dioxide, leaving behind the analyte or its solid residue. The weight of the residue is determined and used to calculate the amount of the analyte. For example, the determination of moisture content in a soil sample involves heating the sample to evaporate the water, and the weight loss is used to determine the moisture content. 3. Combustion gravimetry. In combustion gravimetry, the sample is combusted in the presence of oxygen, and the products of combustion are collected and weighed. This technique is commonly used for 1. Precipitation gravimetry. In precipitation gravimetry, a substance, typically an ion, in the sample reacts with a reagent to form an insoluble precipitate. The precipitate is then isolated, washed, dried, and weighed. The mass of the precipitate is used to calculate the concentration of the analyte. For example, when determining the concentration of chloride ions in a water sample, silver nitrate AgNO is added to the sample, forming silver chloride AgCl. Ag plus Cl minus right pointing arrow. AgCl S Ag. Plus Cl minus. Right pointing arrow AgCl S. After filtration, washing, and drying, the mass of the AgCl precipitate is measured, and the concentration of chloride ions is calculated based on the stoichiometry of the reaction. 2. Volatilization gravimetry. Volatilization gravimetry involves heating the sample to volatilize a component, such as water or carbon dioxide, leaving behind the analyte or its solid residue. The weight of the residue is determined and used to calculate the amount of the analyte. For example, the determination of moisture content in a soil sample involves heating the sample to evaporate the water, and the weight loss is used to determine the moisture content. 3. Combustion gravimetry. In combustion gravimetry, the sample is combusted in the presence of oxygen, and the products of combustion are collected and weighed. This technique is commonly used for determining the carbon content in organic matter or the sulfur content in fuels. After filtration, washing, and drying, the mass of the AgCl precipitate is measured, and the concentration of chloride ions is calculated based on the stoichiometry of the reaction. 2. Volatilization gravimetry. Volatilization gravimetry involves heating the sample to volatilize a component, such as water or carbon dioxide, leaving behind the analyte or its solid residue. The weight of the residue is determined and used to calculate the amount of the analyte. For example, the determination of moisture content in a soil sample involves heating the sample to evaporate the water, and the weight loss is used to determine the moisture content. 3. Combustion gravimetry. In combustion gravimetry, the sample is combusted in the presence of oxygen, and the products of combustion are collected and weighed. This technique is commonly used for determining the carbon content in organic matter or the sulfur content in fuels. For example, the amount of sulfur in a coal sample can be determined by burning the sample in excess oxygen, and the resulting sulfur dioxide is absorbed in a known solution. The mass of sulfur is then calculated based on the amount of absorbed SO. Applications of gravimetry in environmental science. Gravimetric methods. Volatilization gravimetry involves heating the sample to volatilize a component, such as water or carbon dioxide, leaving behind the analyte or its solid residue. The weight of the residue is determined and used to calculate the amount of the analyte. For example, the determination of moisture content in a soil sample involves heating the sample to evaporate the water, and the weight loss is used to determine the moisture content. 3. Combustion gravimetry. In combustion gravimetry, the sample is combusted in the presence of oxygen, and the products of combustion are collected and weighed. This technique is commonly used for determining the carbon content in organic matter or the sulfur content in fuels. 3. Combustion gravimetry. In combustion gravimetry, the sample is combusted in the presence of oxygen, and the products of combustion are collected and weighed. This technique is commonly used for determining the carbon content in organic matter or the sulfur content in fuels. For example, the amount of sulfur in a coal sample can be determined by burning the sample in excess oxygen, and the resulting sulfur dioxide is absorbed in a known solution. The mass of sulfur is then calculated based on the amount of absorbed SO. Applications of gravimetry in environmental science. Gravimetric methods are particularly useful in environmental science for analyzing various substances in water, soil, air, and biological samples. Some common applications of gravimetry in environmental science include water quality analysis. Determination of total suspended solids TSS. Gravimetric analysis is used to measure the amount of suspended Determination of total suspended solids TSS. Gravimetric analysis is used to measure 3. Combustion gravimetry. In combustion gravimetry, the sample is combusted in the presence of oxygen, and the products of combustion are collected and weighed. This technique is commonly used for determining the carbon content in organic matter or the sulfur content in fuels. For example, the amount of sulfur in a coal sample can be determined by burning the sample in excess oxygen, and the resulting sulfur dioxide is absorbed in a known solution. The mass of sulfur is then calculated based on the amount of absorbed SO. Applications of gravimetry in environmental science. Gravimetric methods are particularly useful in environmental science for analyzing various substances in water, soil, air, and biological samples. Some common applications of gravimetry in environmental science include water quality analysis. Determination of total suspended solids TSS. Gravimetric analysis is used to measure the amount of suspended solids in water, which is important for assessing the quality of drinking water, wastewater, and river water. Chloride determination. As mentioned above, gravimetric analysis can be used to measure the chloride content in water, which is important in assessing the salinity of water bodies. Soil analysis. Soil organic matter (SOM). Gravimetric analysis is used to determine the organic content in soils by measuring the weight loss upon ignition, which reflects the loss of organic matter carbon. Soil nutrient content. Gravimetry is used to measure the concentration of essential nutrients such as phosphates, nitrates, and sulfates in soil. Air pollution monitoring. Particulate matter (PM). Gravimetric methods are used to measure the concentration of particulate matter in the air by collecting airborne particles on a filter and weighing the filter before and after exposure. Analysis of pollutants. Gravimetric methods are used to detect pollutants in environmental samples, such as the determination of heavy metals e.g., lead, cadmium, mercury in water and soil. Analysis of wastewater. Total solids determination. Gravimetric analysis helps determine the total solids content in wastewater, which is an important parameter for assessing the treatment efficiency of wastewater treatment plants. Advantages and disadvantages of gravimetry. Advantages. High accuracy and precision. Gravimetry is one of the most accurate and precise analytical techniques, especially when other methods may be affected by interference or instability. Simple and reliable. The method is relatively simple and results. Which of the following substances can be determined by gravimetric analysis in environmental science? A. Phosphate in water. B. Sulfate in air. C. Ammonium in soil. D. Chlorine in wastewater. Answer. A. Phosphate in water. UGC NET 2021. What is the most commonly used reagent in precipitation gravimetry? A. Silver nitrate. B. Sodium hydroxide. C. Hydrochloric acid. D. Potassium permanganate. Answer. A. Silver nitrate. B. Sodium hydroxide. C. Hydrochloric acid. D. Potassium permanganate. Answer. A. Silver nitrate. UGC NET 2020. Conclusion. Gravimetry is a precise and reliable analytical technique used widely in environmental science to determine the concentration of various substances in water, soil, and air. It is particularly valuable in situations where high accuracy is required, such as the analysis of heavy metals, nutrients, suspended solids, and pollutants. Understanding the principles of gravimetry and its various applications will not only enhance your knowledge but also equip you with the tools to approach environmental analysis effectively in the UGC NET JRF Environmental Science exam. You said, bomb calorimetry, ChatGPT said. Bomb calorimetry, UGC NET JRF Environmental Science lecture. Bomb calorimetry is a precise method used to measure the heat of combustion of a sample, particularly in the determination of the energy content of fuels and other substances. It plays an important role in environmental science, particularly in the study of energy production, consumption, and the environmental impact of various fuels. This lecture covers the principles, applications, and techniques of bomb calorimetry, which are crucial for understanding its role in determining the calorific value of materials, especially in the context of biomass and fossil fuels. Principle of bomb calorimetry. The principle of bomb calorimetry is based on the measurement of heat released during the combustion of a sample in a controlled, oxygen-rich environment. The sample is combusted in a sealed container, the bomb, surrounded by water. The heat produced by the combustion of the sample raises the temperature of the surrounding water, and the temperature change is used to calculate the energy released. The bomb calorimeter consists of a strong metal container, the bomb, and ignition system, a calorimeter jacket, and a thermometer. The bomb is filled with oxygen and the sample to be tested, and the bomb calorimetry is a precise method used to measure the heat of combustion of a sample, particularly in the determination of the energy content of fuels and other substances. It plays an important role in environmental science, particularly in the study of energy production, consumption, and the environmental impact of various fuels. This lecture covers the principles, applications, and techniques of bomb calorimetry, which are crucial for understanding its role in determining the calorific value of materials, especially in the context of biomass and fossil fuels. Principle of bomb calorimetry. The principle of bomb calorimetry is based on the measurement of heat released during the combustion of a sample in a controlled, oxygen-rich environment. The sample is combusted in a sealed container, the bomb, surrounded by water. The heat produced by the combustion of the sample raises the temperature of the surrounding water, and the temperature change is used to calculate the energy released. The bomb calorimeter consists of a strong metal container, the bomb, and ignition system, a calorimeter jacket, and a thermometer. The bomb is filled with oxygen and the sample to be tested, and the entire system is submerged in a known quantity of water. After igniting the sample, the heat produced during the bomb calorimeter consists of a strong metal container, the bomb, and ignition system, a calorimeter jacket, and a thermometer. The bomb is filled with oxygen and the sample to be tested, and the entire system is submerged in a known quantity of water. After igniting the sample, the heat produced during the combustion is absorbed by the surrounding water, and the change in temperature is recorded. This temperature change allows for the calculation of the energy released during combustion. Components of a bomb calorimeter. Bomb, a strong metal vessel designed to withstand high pressure, which is used to hold the sample in oxygen. Sample, the material whose calorific value is being measured, e.g., coal, biomass, oil, food. Oxygen, pure oxygen is added to the bomb to ensure complete combustion of the sample. Ignition system, a small electrical ignition system is used to ignite the sample once the bomb is sealed. Calorimeter jacket, the bomb is placed in an insulated water jacket to minimize heat loss. Thermometer, the thermometer measures the change in temperature of the surrounding water. Stirrer, ensures uniform distribution of heat in the water during the experiment. Procedure of bomb calorimetry. Preparation of the sample, a known weight of the sample is accurately weighed. The sample should be finely powdered to ensure complete combustion. Placement in the bomb, the sample is placed in a small crucible inside the bomb, and a known amount of oxygen is added. Sealing the bomb is securely sealed to prevent the escape of gases during combustion. Placement in the calorimeter, the bomb is placed in a calorimeter jacket, which is filled with a known amount of water. The thermometer is placed in the water to monitor the temperature change. Ignition, an ignition system is used to ignite the sample, initiating combustion. The heat produced by combustion is absorbed by the water in the jacket. Measurement of temperature change, as the sample burns, the temperature of the water increases. The change in temperature is recorded during the reaction. Calculation of energy released, the amount of heat released by the sample is calculated using the temperature change of the water and the known heat capacity of the water. The energy released per gram of sample is determined. The energy released during combustion is typically expressed in calories cal or joules J. The standard unit for measuring the calorific value is kilocalories per gram, or kilojoules per gram, kilojoule per gram. Calculation of heat of combustion. The heat of combustion is calculated using the following equation. Q equals m times delta T times C. Q equals m times delta T times C, where Q equals heat released calories or joules. M equals mass of water G. Delta T equals change in temperature degree C or K. C equals heat capacity of water. Specific heat, generally taken as 1 calorie per gram degree C or 4.184 J per gram degree C. Once the heat released is calculated, the calorific value of the sample is determined by dividing the heat released by the weight of the sample. Calorific value kilocalorie per gram equals Q. Mass of sample G. Calorific value kilocalorie per gram equals mass of sample GQ. Applications of bomb calorimetry in environmental science. Bomb calorimetry plays an essential role in various fields of environmental science, particularly in the study of energy production and its impact on the environment. Some of its key applications include determination of fuel quality. Bomb calorimetry is widely used to measure the energy content of fuels, including coal, oil, natural gas, and biofuels. This information is critical for evaluating fuel efficiency and selecting appropriate fuels for energy generation. Biomass energy. Bomb calorimetry helps determine the calorific value of biomass wood, agricultural residues, waste which is used in the generation of renewable energy. Biomass fuels are considered a sustainable energy source, and understanding their energy content helps optimize energy production. Waste to energy conversion. The calorific value of various types of waste materials, e.g., municipal solid waste, plastics, organic waste, can be determined.